Thank you, Mark. On behalf of my co-investigators, I have the privilege of presenting key takeaways from the RGX 314 gene therapy trial in previously treated wet AMD. My financial disclosures are shown here. I serve as a scientific advisor to REGENXBIO. RGX 314 utilizes a proprietary gene delivery platform that's hypothesized to deliver longer and higher protein expression with a lower immune response. The AAV8 vector is encoded to deliver a gene that is for anti-VEGF monoclonal antibody fragment. The trial enrolled previously treated neovascular AMD patients who demonstrated a high need for anti-VEGF therapy. They also had to demonstrate a level of response to anti-VEGF therapy at screening. They then received the RGX 314 gene therapy subretinally and were followed monthly for safety and for need for rescue therapy. 42 subjects were enrolled across 5 dose cohorts. Anti-VEGF retreatment was allowed for any fluid or disease activity to include increased, new, or persistent fluid, vision loss, or new ocular hemorrhage. The demographics were typical of a cohort for wet AMD patients. You'll note that serum neutralizing antibodies were taken, but this was not an exclusion criteria. Almost 50% of patients demonstrated these, which is consistent with the literature. The average annualized injections prior to entry ranged from 7 to 10 and demonstrated the high need for frequent anti-VEGF therapy. RGX 314 was well tolerated. There were no drug-related serious adverse events and no observed clinically determined immune responses. 2 deaths were unrelated to RGX 314, and there were 2 ocular procedure-related serious adverse events: a peripheral retinal detachment, which was successfully repaired, and a post-aqueous sample collection endophthalmitis. There was a dose-dependent increase in RGX 314 protein observed across the cohorts. You see cohort 4 and 5 here, which has not been previously presented. When we look at cohort 3 on the right, we see sustained visual and anatomic outcomes over 18 months, despite relatively few injections. 3 of the 6 patients in cohort 3 were injection-free at 18 months. On the left, you see sustained protein levels over 1 year with stable visual acuity and central retinal thickness. This is cohort 4. These patients were stable to improve with respect to vision and anatomy. 5 of the 12 patients were injection-free at 6 months. 2 patients had an incomplete response and required monthly anti-VEGF. Here's cohort 5, also with stable to improve vision and anatomy. 9 of 12 patients were injection-free at 5 to 6 months. Here we see cohort 4 and cohort 5 with the subjects that were injection-free at 5 to 6 months, and you see stable to improve visual acuity and good central retinal thickness. This is a schematic showing the need for injections in cohort 5 in the year prior to delivery of RGX 314. And on the right, you can see the rescue injections after the surgery. And you can see there were very few injections, and most of these were to 1 patient. This is one of the patients from cohort 5. This is a patient who required 13 injections in the year prior to enrollment. You can see persistent fluid throughout. And after delivery of the RGX 314, no rescue injections were needed through 5 months. This is a second patient, 8 injections needed prior to enrollment, and after RGX 314 was delivered, no rescue treatment through 6 months. Based on these findings, REGENXBIO is going to look at a phase 2b study in wet AMD, which will be starting by the end of the year, an IND for diabetic retinopathy. They will also be expanding to evaluate suprachoroidal delivery using Clearside's microinjector. So in summary, subretinal RGX 314 was well tolerated with dose-dependent increases in ocular protein, and in cohort 5, there was a high clinical response with 3/4 of the patients injection-free at 5 to 6 months. Thank you.